guys, and welcome to IB Online. Javed Khan Afridi here, and you are watching AS Level Biology. It's the time to discuss the most important step of mitosis, and that is called metaphase. In the previous video, I told you about the detail regarding the prophase, the longest phase, and now we are in metaphase, and in metaphase spindle formation is going to be completed, and each spindle fiber will be attached to the centromere of each chromosome and will drag them to bring them all the chromosome to be aligned on the metaphase plate. How? Let's to discuss now in detail. Let's see the structure here, and this structure is going to tell you these are chromosomes. We have 4 chromosomes here: chromosome 1, chromosome 2, chromosome 3, chromosome 4. The nuclear membrane is already broken down. Initially, the nuclear membrane was present, but I told you that the nuclear membrane has been disintegrated, fragmented, and then disappeared in the prophase. In the prophase, the nucleus was disappeared, the nucleolus was also disappeared. So this is also disappeared, the nuclear membrane was also disappeared. These two structures are not here, and now the DNA and the chromosomes now, the DNA in the form of chromosome now naked, as they don't have any nuclear membrane. Now you can see here the centrosome or centriole, one here and one here, and these are the pole. These are the pole. I mean, if you mix the nucleus like in this way, then these are the poles, and this is the equator. Just like in globe, you can see the globe has, I mean, if I'm making the nucleus like in this way, then these are the poles of the nucleus, and this is the equator. So this is the equator. Equator is just the equal distance from the centrosome. The centrosome and both the centrosome must be at an equal distance from the equator, just the middle part. If the nuclear membrane were present, then it was the center of the nucleus actually, but there's no nuclear membrane now. You can see that no one of the chromosome on the equator. They were just on the right and left side of the equator. So what's going on in metaphase? The first important step in metaphase to bring these chromosomes at the metaphase plate, and they should be aligned vertically together. These chromosomes should be like in this form. They will be aligned in this way. I mean, the chromosome must be like aligned here in this way at the equator. How many chromosomes we have? 4. So all these 4 should be arranged in this form. I mean, they should be this will be moved toward the left, it will toward the right, and bring them on the equator. So how they will come on the equator? They will be pull apart. So for pulling, we need some types of protein filaments to bind it and just pull it and drag it toward the equator. Of course, these are spindle fibers, and these spindle fibers should be reached here, and then they will pull apart the chromosome. So like I told you that initially the spindle fibers are also formed to some extent, but they are not complete now. Like I told you that the spindle fibers have been formed, but it remains continue and not completed now. Similarly, here also the spindle fibers have been formed but still remain continue and not completed from both the sides. This was the case in prophase, I told you, and they are protending toward the respective chromosomes in prophase. This was a step in prophase, and I told you that some of them already attached, but they are not only here. And keep in mind that the number of spindle fiber are never equal to the number of chromosomes. Here we have 4 chromosomes and we have shown 4 spindle fiber, but this is not actually the case. We have so many spindle fibers which are moving from one pole and extended to the other pole without attaching any one of the chromosome. Like there may be some of the spindle fibers which are just passing from here without touching the chromosomes. So many spindle fibers passing them. They are also the dimers. They are also made up of the dimers and remains continue, but they are not joining the chromosomes. So there's a lot of spindle fibers which are overlapping one another, but they do not reach to the chromosomes here in the same way. These are the dimers, the spindle fibers. They're not just touch one another, they're just overlapping on one another, and these are called the spindle, overlapping spindle. These are called overlapping spindle. They're never attached to the chromosomes, but just crossing one another. Similarly, the other one is also, they're overlapping on one another. However, these, the inner one of the spindle fiber, they are growing toward their respective centromere. So where the centromere, just going to mention here, the centromere is the region like this is the centromere, here is the centromere, here is the centromere, and here is the centromere, where the dimer is going to be attached with the chromosome. Now, how these spindle fiber reach to their respective centromere? The dimer should be added or should be removed. There are free dimer which join with them and extending the growth of the dimer. They are moving, and they are moving in the same pattern, like I'm just going to show it like the case here. Just make one of the spindle fiber here. The spindle fiber have been extended from the centrosome and reach to the spindle, reach to the centromere and bind with the centromere. This is one of the dimer extending from one of the centrosome. So you say, in this case, the dimer have been added. We have added the dimer. Once the dimer have been added one by one, it's just growing like a chain. So a chain of dimer is formed only when you are adding some dimer. So they are not actually stretching or contracting. It's just not like a rubber or like a muscle. It's a muscle, but like types of contractile fibers. But they are actually formed by some units of the dimer to be formed and extending. Similarly, in the other side, also we have the extension, like they are extending from both the side, it's extending. So they are attached. This chromosome has been bound from both the side by the dimers, by the spindle fiber. One of the spindle fiber from one of the pole, from one of the centrosome, and the other from the other centrosome. The same is the case here with the other one. If you see the dimer here, sorry. You can see here, the dimer should be like also attached with this one. So here again, we have extension of dimer from this side. The dimers are going to attach with this one. And similarly, further extension of the dimer from this side as well. So there we continue. One of the fibers should be longer one and one should be the shorter one. The one which is closer to the equator, they have the shorter one, and the one which is so away from the equator, they have a longer fiber. So here, this one, in this case, this is the longer one, and this one is the shorter one. Joined together. Similarly, the extension remain continue in the other one as well. Here, like, once again, the extension remain continue with this one. It's also the growing one. So the dimer are still growing and until it reached to this centromere. It's reached here. Similarly, the other extension from the other side remains continue in the same format. Here, like, it will continue to bind with the other part of the centromere. Again, extension growing of the dimer to make the spindle fibers. And similarly, the last one, extension of dimer. So you can see here the extension. And from the other side as well. The centromere is attracted from both sides. Now the spindle fiber have been completed, and we also have the overlapping one. These are the overlapping one without binding to any one of the chromosome, and these are the spindle fibers which are the attached to the chromosomes. Now what you have observed here, that if I'm talking about this centromere, I mean this one, and also this one, centromere. So from both the centrosome, not centromere, I'm so sorry, centrosome. From both the centrosome, the dimer were growing. I mean, growing means the chain was elongating, not shortening. I mean, from both the side, the dimer were adding. The dimer have been added here, also here. So the chain grows. Similarly, the dimer have been added here, addition of dimer, and it is also addition here, and the dimer have been grown from both the side. This is called the plus end. I mean, if I'm telling you that this side, when you are moving the dimer from in this format, so we say that this is the plus end, because the dimer have been added. This is also the plus one, this is also the plus one, because in both the side the dimer have been adding plus from both the side. Similarly, this is also the plus one, this is also the plus one, and the dimer have been added from both the side. When you are removing the dimer, that's short minus if you remove the dimer, but we are not removing the dimer, we are just adding from both the side, so they reach and attach to the chromosome, to the centromere. Now, why we are doing all these things? Our aim is to bring these chromosomes on the equator. So if you want to pull this chromosome and bring it to here, what should you do? You just have to remove the dimer from here. If you remove the dimer, you're just making this chain shortening. Once the dimer should be removed from the middle part here, it just like become overlaps like in this way. This part is removed out, the chain becomes shortened, so it makes a stain, it makes a pulling force on the centromere, pulling this chromosome here. But if this one is the removing one, the chain becomes shortening and the chromosome is pulling apart. Now there's a risk that the centromere might be broken down, because if one dimer is pulling here and the other one is pulling here, then the chromosome will be broken down. But it's not the case. The dimer is removed from this side. However, still the dimer is adding from this side. So if you are removing the dimer from one side, the chain is shortening, but the other side become lengthening. It means you are pulling the chromosome from one side and the other side is pushing one, because from one side we are pushing the chromosome. By the arrival of new dimer, you are elongating the chain. But on the right side here, you are shortening the chain. So if you are shortening the chain, you are just pulling the chromosome. On the other, from the back side, you are adding more dimers, so you are pushing one. In this way, the chromosome will be brought about toward the center. Like in this way, this chromosome will be brought to here. So if it is brought to here, it means this chain have a distance. So we need some more dimer to grow here. That's why we say this side should still be a plus side. You have to add some more dimer to push it here. However, you can see that these dimer. You can see like these dimer are extra. They should be removed. They should be removed from the chain. So when you remove these dimer from the chain, now it will become like, see, the dimer should be removed. Now look at this one. What's going on here? Actually, we just have removed the dimer from this side and we have added dimer from this side. So in this case, that this side was still the plus one, but this was become the negative one because you are removing dimer from one side. In this way, the chromosome have been arrived here. Similarly, now what will go in the lower one case? Now look at this and focus here. Which strand is the longer one? Of course, the red one. and the blue one is the shorter one. You can see this is the shorter one and this is the longer one, because we need this chromosome to bring backward. Like this one we have brought in here, it should be here. So there are greater number of dimers. So from here we will remove the dimer. So when you are removing the dimer, you're just pulling the chromosome here, and you have to add dimer here to push this one here. So exactly what's going on, like we are pushing this chromosome to here. You can see here. So if you are moving here, you need some more dimer to be completed. So exactly what should we do? We have to add more dimer here, more dimer. We need some more. And you have to remove the previous one from this side. So we just have to remove the dimer, and we have add on this side now oppositely. So in this case, we have removed the dimer from this side, and we have added dimer in this side, just opposite. Like here we have removed and add, and in the lower case we have add and removed to bring it on the equator of the spindle. Again, the same case here. Now, which one is the longer one? This one. So again, here will be remove the dimer, and here we have to add the dimer to bring these chromosomes here. So this side will become negative and still remain positive, the addition and then removal. In the same case, here you have to bring these chromosomes to here. So if you want to bring here, yes, you have to need some more dimer to be added. Look at here. So if you put it here, so you have to first remove these dimer. And then you have to pull it like at the original form. It should be like this. And let me to finish to clear all these massive unnecessary structures from here. Now you can see that the dimer should be completed, like the red one, to grow, to shorten, and it has to become added, and the chromosomes will be added to here. Similarly, in this here the case, the same the case with here. So we just have to increase the number of dimer here and decrease the number of dimer here to pulling here the chromosome. So this chromosome will be pull apart again in the same format, and we will pulling these chromosomes to here. Oops, let me to draw it again here. Just have to clear this one. So you can see you just have to add some dimer here. Sorry. So you have to grown up this one. You have to add more dimer. And of course, you have removed the red one, which have removed from that part. So this one, the negative, and this was the positive. So this is how we have aligned all the chromosomes at the equator by just shortening and lengthening the dimer. Now this is the story how this is working. Now this addition of dimer and removal of dimer is controlled by two types of enzymes which are working to add on one side and remove the dimer from the other side. Then let's see the story here in the metaphase. So what's going on in the metaphase in the detail now? During metaphase, the spindle apparatus develops fully. This is one of the key points. The spindle fiber completely fully developed. Most of the spindle fibers do not attach to chromosome. I mean, they are talking about the overlapping one, but extends from one pole to the other. Now, what's the benefit of these fibers? We didn't mention what's the role. They will play their role in anaphase. In the later stage, they will play a very important role. These fibers provide a structure, and with the fiber that are attached to the chromosome can function and give spindle its characteristic shape. How they will allow the spindle fiber which are attached to the chromosome a specific function, that will be discussed in the later stage of mitosis, that is anaphase. Now, during metaphase, the chromosome become aligned. They are aligned at the equator. They just make a row, just a column. Sorry, not a row, it's a column. They will be arranged in the form of a column on the equator along the center of the spindle. This is an important stage in mitosis as it determines how the chromatids will segregate it later on. Why we are aligning the chromosomes on the spindle? Because we want to easily separate their respective sister chromatids on both the side of the pole. If you want to chop some potato sticks, so first there are a lot of potato strips, and they're spreading here and over there. You just have to make the strips align, and just make a cut on the knife. So just makes all the strips in the same form, in the same distance and the same length. That's why we want the chromosome, the chromosome with the two sister chromatids to be aligned on the equator. That is, make a cut so that equal number of chromatids have been segregated, moved to the opposite pole of the cell, and once cytokinesis takes place, so each cell will contain equal number of DNA, equal number of the sister chromatids, because there you have equally split them. Now in metaphase, I'm explaining the assembly of the chromatids here. Role of spindle fibers in the separation of chromatids. Spindle fibers have three destinations, some attached to the kinetochore. What is kinetochore? Kinetochore is a kind of protein which overlap the centromere. So the chromosomes not directly attached to the centromere, they are attached to the kinetochore. Let me to show the structure of kinetochore might be here. If you see here, this kinetochore, yes, here the structure. You can see this is the kinetochore structure. These are the spindle fibers which are attached. This is the centromere. This part is the centromere. And these are one of the sister chromatids. This is another sister chromatid. The entire one is one of the chromosome. And one chromosome have one centromere. The centromere is overlapped by the kinetochore. So the spindle fiber not directly attached to the centromere, ratherly they are attached to the kinetochore. Kinetochore is a kind of protein. I'll tell you in the next video. So here the function of the kinetochore is mentioned. That's why I told you. While those growing from opposite centrosome point to the other kinetochore of the same chromosomes, right, still continue growing from the two centrosomes until they extend between each other in a region of overlap. So there are two types of spindle fiber. I told you some overlapping with one another without touching any of the centromere, and they were attached. Now all these groups of the spindle fiber participate in the assembly of chromosome. Assembly means they have to assemble them on the equator at the metaphase plate. Metaphase plate and equator are the same thing. Metaphase plate or equator, they're the same thing. And separation of the chromosome at anaphase. So assembly of chromosome at metaphase plate. Let's see the diagram here. Look at this diagram. This is one of the chromosome. And we want to bring this chromosome at the metaphase plate. This is the future metaphase plate. The dots show the metaphase plate, and this chromosome should be brought in here. Now this is one centrosome, this is another centrosome, and you can see these are the dimers which have been formed. So this one, the longer one, because it's far away from the chromosome is closer to this centrosome and it's far away from this one. You can see this spindle fiber is quite longer one, and this is quite shorter one. Now what will go in, where the dimer should be added? The dimer should be added here to lengthen it. And since this is the longer one, so dimer should be removed. So it's pull the chromosome to here. And you can see this is the plus side because the dimer should be added to here from negative to positive. But this is moving from positive to negative because you are decreasing the dimer. So if you are moving from positive to negative, you are just removing the dimer and making the spindle fiber to be shortened. Once it's shortened, it's pulling the chromosomes to here. And when it's adding, so it's just pushing it and bringing toward the metaphase plate. Now, these are the microtubules I told you, and this is the tubulin. Each unit is the tubulin, and this is the kinetochore I told you. Sorry. Dynein and kinesins are the two enzymes which are working. So dynein is, this is kinesin, sorry. Kinesin is used in the addition of dimer. They are involved in the addition of dimer, lengthening. They involve in the lengthening. But dynein is involved in the shortening as it removes the dimer. So if one is shortening and the other one is lengthening, it brings the chromosome here at this stage. And that's why we say here that the microtubules attached to the opposite side of the chromosome shrink. Shrink means shorten or grow until they are of equal length. So they grew and become the equal length, means you can see the length will become equal. If you bring it to here, then the length of both the strands will become equal as you are in the center. As I show the chromosome here, you can see now the length of the fibers become equal. This and this have the same length. When the length become equal, the chromosome will be aligned in the center. But initially this chromosome was located where? This was located here. And in that case, this was the shorter strand and this was the longer strand. But we have removed the dimer from here to become shortened, and we have added the dimer to become it lengthened, and that's why the chromosome have brought in here at the equator. Similarly, once the metaphase has been completed, at the end of metaphase, you can see that all the chromosomes are aligned at the equator, and the spindle fiber from both the sides should be of the same length. They have the same length actually. So here it says that what will happen at the end is say that the microtubules attached to the opposite of the cell shrink or grow until they are of equal length. Microtubules motor, means enzymes attached to the kinetochores, move them toward the minus end of the shrinking microtubule, dynein. This is the dynein. Shrinking means removal of dimer. Minus end means is shortening, and toward the plus end, lengthening microtubule, which is the kinesin, because here the dimers have been added. So this chromosome will be arrive here at the center. So we have aligned all the chromosomes at the center of the metaphase plate. This is what's going on in the process of metaphase. And what's going on in the anaphase now? This is very important. Anaphase is just a shortening of the chromosomes of both the spindle fiber to split the chromosomes, and they will be moved to their respective poles. Now here we have the region of. Now this is the case. You can see this diagram. Let me just show you. See this diagram. This diagram is the end of, prophase, and now we have to start the anaphase. So what's going on in the anaphase? What's going on in the anaphase? These are the overlapping spindle fiber I told you initially. This is one of the spindle fiber, another one overlapping. This is also the overlapping. This is also the overlapping one. So you can see the region of overlap is shown by the shaded area. Here, 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 and here. So we have 4 overlapping spindle fiber which are not attached to chromosomes, and we have 3 normal spindle fiber which are attached to the chromosomes, as I told you initially. Once they overlap with one another, in the region of overlap, you can see these blue lines. They show the hydrogen bonding. So the 2 spindle fibers, one moving from one pole, the other from the other pole, they just overlap one another, and at their end, hydrogen bonds are formed to make them like a single fiber. Now it's behaved like both of the pole are now connected with one another, although they are not connected with one fiber. But the pole of both the fibers have been joined together. If you seal the end, so it means that the fiber directly connect both the centrosomes with one another. This is one of the centrosome, this is one of the other centrosome. So both the centrosome have been connected with the help of the fiber. So it behaves like the one of the spindle fiber to be collecting these two. The same here is undergoing here, is one here, but since they are joined with the hydrogen bonds, it makes like a single fiber to be in between them. So the poles, the centrosomes are also directly connected with one another through these 4. This is one of the fiber, this is another one, this is another one, and this is another one. So these 4 fibers connect the centrosomes with one another. And these 3 fibers are connecting with the chromosomes. Now the story begins here. What will happen if we are adding dimers in the overlapping spindle fiber from both the sides? If you are adding dimer here and you're also adding dimer in both the sides, it means you are making lengthening of this side and also lengthening of this side. From both the side, we are lengthening the fibers. It's the same. So if you are lengthening the fiber, they just pull the poles to the opposite way. I'm just giving you an example. If there are two students and in between the students, we have a flexible rod, like the rod of an umbrella. If you just push the button, the rod become lengthened. They have a spring in between them. So there's a short rod in between the two students and the rod become lengthened. So both the students will be moved away from one another because the distance between the students will be increased. In the same way, if you lengthen this spindle fiber, they will just pull the centrosome far away from one another. So the distance between the centrosomes will be increased, pushing apart the centrosome. But keep in mind, if you are moving this centrosome here and this centrosome here, now these centrosomes are also connected with the other wire with the centrosome. So they're just pulling one of the chromosome here, the other chromosome here. So extension of the overlapping spindle fiber causing the shortening of the other fibers which are connected to the chromosome. So of course, if you are lengthening these one, they will split the spindle fiber which are attached to the chromosomes. So two things are going at the same time: lengthening of the overlapping spindle fiber and shortening of the spindle fibers which are attached to the chromosome. If I say, like I'm increasing, I'm adding dimer, and similarly in all the overlapping fiber we are adding dimer, so we are just increasing its length. In all these four spindle fiber are increasing in length. So these spindle fiber will be pushed away from one another, like their distance will be increased and they will moving like this pole will be like, just check the color here. So it will be like here, the pole have been increased and the other one have been increased to here. So their shape, the spindle fiber should be like now in this form. Just have to increase the length. They're so far away from one another. So the chromosomes which are in the middle, of course, they will become broken down because their length cannot be increased. The overlapping one have been increased by the addition of dimer, but we are not adding any dimer in those spindle fiber which are attached to the chromosome. So if you are pulling the lengthening, the one which are attached to the chromosome will become shortened and they will be broken down. So I say if I remove the dimer from these spindle from both the side, if you are removing shortening taking place from both the side, so you're just splitting the chromosomes, pulling. The one of the chromatid will be pulled to the right and the other toward the left. So you're just putting a stain on the chromosomes centromere to break them. And the other force is because of the lengthening of the spindle fibers which are overlapping. So lengthening of the overlapping fiber and shortening of those which are attached to the chromosomes on both the side break the chromosomes and they remove the chromosomes from one another, break them. So the shape will be now modified. How the shape will be modified, let me to draw it now. So here now, I'm just going to copy this shape once again and just making changes in between the diagram. Here, you can see what will going on now. Here, at the end, let me to finish this one as well. The structures here. What will going on? If the dimer has been removed, so these chromosomes will be splitting down and shortening taking place. So this will be shortened. Now one of the chromosome will be attached here and the other one here. They become separated because of the shortening of the the spindle fiber. And this structure will move backward. Like in this way it's moving, so just breaking the chromosomes. And similarly this one is also moving like here, so they're just breaking the chromosomes, but the overlapping spindle fiber will be still growing as they will not be broken down like this one. As let me to clear it, this one is still remains continue as it has been grown, and the hydrogen bonds still in between them. So the overlapping one. Similarly, the overlapping one of here, it should also be remain continue. So the hydrogen bond still present here and become in lengthen. Similarly, this one also remains continue. They're still joined together with one another. Sorry, some more clearer. So we have the hydrogen bond here, still remain continue. And finally, the last one, it still remain continue. And we have the region of overlap and the hydrogen bonds here. So they are still continue with one another. However, the chromosomes here have been broken down. This one is attached here. Similarly, here is, sorry, they have been separated from one another. Similarly, in this structure, here the one is attached here, the other one is attached here. So this has been also split down. In the same way, you can see this one is also broken down. And now one of the chromosome is reached here, and the other has been reached here, and it is split down. So now you can see the shape became here, and it's become further shortening taking place here, further shortening taking place here, and still this chromosome will be attached to, oops, sorry, let me draw it the new one. So here it should be attached, and similarly the black one also to be attached here. So you can see in the same way here, shortening remains continue, and here we have the purple one, like here, chromosomes here, and then the black one, of course. So we have split down the chromosomes from one another. You can see the shape of the spindle fiber now become elongated. The overlapping spindle fiber are lengthening from both the sides, but the spindle fiber which are attached with the chromosome have been split down the centromere, half of the chromatids at one side of the pole and half on the other side. So now this was the shape in prophase at the end of prophase, and now this is the shape at the end of anaphase. So what's going on in the anaphase? Let me to explain now. What's going on in the anaphase? In the anaphase is the shortest phase of mitosis. For each chromosome, it involves the splitting of the centromeres due to the shortening of the fibers on both the sides, the separation of the two sister chromatids from one another, and the movement of the sister chromatid to the opposite pole of the cell. So these are the three consecutive and characteristic points of anaphase: splitting of the centromere, separation of the two centromere from each other, separation of the two chromatids from one another, and arrival of each chromatid on their respective pole. So half of the chromatids now at one side of the pole and half of the chromosomes on the other side of the pole, because each chromosome was initially made up of two DNA molecules. So one of the DNA molecule in the form of chromatid will be on one pole and the other one on the other one. So how this separation takes place now, let's to discuss it now. The sister kinetochore separate. Kinetochore means, you can say, the centromere separate and carrying their attached chromatid move along the microtubule, while the microtubule themselves shorten, probably at both the ends. The shortening of the microtubule and both the sides involved to split the centromere. Now, what's going in the overlapping one? The overlapping spindle fiber move past each other, pushing the poles further apart. So once they are enlarging, they are lengthening, they're just pushing the centrosome at the back, and they are moving far away from one another. The spindle fiber pulls the two chromatids from each chromosome to the opposite pole of the cell. As the spindle fibers are attached to the centromere forms the leading part of each chromosome. So if you're just pulling my finger, look at my finger here. I'm just pulling the finger from back. So it will look like this one, the opposite side, just like a V-shaped structure. Now, if I just pulling it from here, from the center, it will be like this one. If you're moving from that side, then it will move like on the back side. If you have a thread, and just keep the thread from the middle, it's just moving like in this one. So the air fraction will move its position like in opposite V. That's why we had made the chromosomes. So if I'm giving you an example, if this is one of the chromosomes and you're just pushing in this form, so its shape will be like this way. But if you are pushing the chromosome from this side, then its shape will be like this one. So the case here, which I have shown the diagram, why its shape is like this, because it was pushed from back, and these were from pushing from this side, so they are moving like in this way. The axis will be in the opposite way. They show the opposite movement. They show the opposite shape against their movement. So the same thing here. As the spindle fibers are attached to the centromere form the leading part of each chromosome. If the centromere is near the center of the chromosome, then the chromosome has a V-shaped structure. If a chromosome just give me an example, like this is one of the chromosome, and the centromere is in the middle, then after separation, they will just make a V-shaped structure. This is one of the V, this is other of the V. But if the chromosome is like in this way, that the centromere is just at one side, then after splitting, they will make a shape like this one. However, sometimes the centromere at the lower side, and this is one of the chromosomes. So after their separation, they will be like this way. They will become like it this way. They will be just making their shape. So as it's pulled to the pole, at the end of anaphase, there are two groups of chromosomes now, one at each pole of the cell. The two groups of the chromosome form contain one from each pair of sister chromatids. So initially, the sister chromatids now become the chromosome. I mean, they were like in this form at the spindle, at the metaphase plate, but after the separation in anaphase, one is like in this way, the other one is in this way. Now, in this case, each of these two is called chromatid, but after separation, they are now known as chromosomes because each of them have its own centromere, and centromere represents a chromosome. Here we have only one centrosome, so the entire structure is one chromosome. And now we have two chromosomes because each sister chromatid is also considered to be a chromosome. Now, they are the two groups of the chromosome. They say chromosomes, not say chromatids. Formed, contain one from each pair of sister chromatids. Therefore, the two groups of chromosomes carry the same genetic information, they have the same DNA, and they are genetically identical. This is what's going on in the anaphase, which I have explained in detail. Hopefully, you have got the idea what's going on in the anaphase. In the next video, I'll tell you about the telophase, which is the reverse of the prophase, and then cytokinesis. See you in the next video.